Hi everyone, my name is Sion, and I am an assistant instructor for the School of Biosciences at Georgia Tech, and this is a really quick review of the metabolism of animals and plants, so the processes of cellular respiration and photosynthesis. So these are quite a lot of lectures and a lot of learning objectives to cover, and so just try and lock in and commit it to your brain. That way you have this memorized come exam time. So we're going to go ahead and get started with metabolism in animals, so the process of cellular respiration. And we see here that this is the overall reaction where we're taking glucose, a 6-carbon molecule, a sugar specifically, we're combining it with oxygen and releasing carbon dioxide, water, and a bunch of ATP. So that's the last time you'll ever see this equation because we think about it in terms of these 4 reactions: glycolysis, pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation, which you should know consists of the electron transport chain, which is pumping a proton, as well as chemiosmosis, which is the use of the proton gradient to spin ATP synthase, allowing us to generate ATP. Okay, so the entire idea is that we're taking food, we're running these 4 processes, and we're getting the energy from food to perform oxidative phosphorylation. And you should kind of have an intuitive sense for that already. So I like to think about metabolism and also the fact that metabolism is just a series of redox reactions. So for a redox reaction, we have one thing that's losing electrons and another that's gaining electrons. We see this here where we have sodium, which is neutral, becoming sodium, the positive cation, and in doing so, it has become 1 electron more positive, so it is oxidized. It has lost an electron. We see this happen a lot in our metabolism. One of my favorite things to say is that NADH reduces the electron transport chain, because remember that NADH is carrying 2 electrons, and so it gives those electrons to the electron transport chain, allowing a proton to be pumped. And so the electron transport chain is reduced because it has gained electrons. Meanwhile, NADH is oxidized because it has lost electrons and become NAD+. So when I say oxidized and reduced throughout this video, hopefully you'll understand what those terms mean, because metabolism is just literally transferring electrons to the electron transport chain. And we should see this here where what we're doing is oxidizing food. That's the point of metabolism, because oxidizing food releases electrons. So we are releasing electrons to the electron transport chain in order to power what process? Oxidative phosphorylation. That's what's happening here. And we're carrying those electrons to the ETC to perform oxidative phosphorylation via NADH, our electron carrier. So our 3 metabolic processes, which you can see here, glycolysis, pyruvate oxidation, and the citric acid cycle, are all releasing NADH to be carried to the electron transport chain. So we're oxidizing food to release those electrons. Remember, oxidation is loss of electrons. So all those electrons are going to be carried to the ETC via NADH, and that's how we're generating the majority of our ATP. So the first process in that is glycolysis, and I'm going to have the completed chart for these processes linked in the description. It's also on Canvas, so if you haven't filled it out by now would be a great time to do that. Just fill it in as we go. It's a really good idea to have this memorized, like genuinely, like pay attention so you get this diagram filled in, you know the inputs and outputs. So our first step is glycolysis. And glycolysis, we're taking glucose, which is 6 carbons, and we're breaking it down into 2 3-carbon molecules. So we haven't really changed it too much, except when we break that one bond, we release 2 electrons. And so those electrons are going to be carried away via NADH, our electron carrier. Similarly, we produce 4 ATP as a result of substrate level phosphorylation, which means we require ADP as an input. And specifically for glycolysis, we also use some ATP as an input. So it's both an input and an output for glycolysis. I did say in lecture that you don't have to memorize specific numbers, and you don't, but I included them here for ATP and ADP just to make it make a little bit more sense. Glycolysis requires an energy investment in the form of 2 ATP, but we net, or we produce 4 ATP, so we net 2 ATP. So just understanding that for glycolysis, we have ATP as both an input as well as an output. And glycolysis is going to occur in the cytoplasm. We see this right here in the cytosol. And what happens is that we're going to take these pyruvate and we're going to export them to the mitochondria. So they're going to pass through the mitochondrial membrane, and we perform pyruvate oxidation. So we take those pyruvates and we turn them into acetyl-CoA. So if we attach this CoA group, that would obviously mean that we have CoA as an input. And if we release NADH, we have NAD plus as an input, and we also release some CO2. And this is going to happen in the mitochondrial matrix. So we've entered inside the mitochondria. So again, just try and get that chart filled in. So then we're going to take this acetyl-CoA and run the citric acid cycle. So in my mind, the major product of the citric acid cycle is lots and lots of NADH. So in addition to producing NADH, we produce FADH, which for all intents and purposes can be functionally the same as NADH. So similarly, if we produce NADH as an output, we need NAD plus as an input, and the counterpart to that for FADH is FAD. We also produce some ATP as a result of substrate level phosphorylation, which means we require ADP as an input. And again, this is happening in the mitochondrial matrix. So those were the 3 major oxidation steps of food. So we're taking that glucose, we're oxidizing away its electrons, carrying it away in the form of NADH, and now that we have a bunch of it, what are we going to do? Well, we're going to perform oxidative phosphorylation. So I have that here as the last step in your chart. So it's when we, again, reduce the ETC, we give our electrons to oxygen, the terminal electron acceptor, and that is reduced into water, and NADH is oxidized into NAD+. So we've given away our electrons to the ETC and become NAD+. So I call this step the oxidative phosphorylation step, the ETC step. It's just there on your chart to see what's happening to the NADH we produced from the citric acid cycle. And we see oxidative phosphorylation in the electron transport chain happen graphically here, where we have our NADH. It's giving its electrons to oxygen, the terminal electron acceptor, and it's being oxidized itself to NAD+, and we're pumping those protons and allowing ATP synthase to spin. In the form of a map, how I've put this all together is that we have glycolysis occurring in the cytoplasm. We take that pyruvate, we export it into the mitochondria, we oxidize it, and then we run the citric acid cycle, produce a bunch of NADH. That NADH is going to go reduce the ETC and become oxidized to NAD+, which is our first input, so the cycle can repeat itself. So again, just a different form of that graphic, just not made by me, so it's not as good in my opinion. But again, we have this last step of oxidative phosphorylation occurring here, and we're taking glucose, we're taking our sugar, we're releasing our electrons in the form of NADH, and we're carrying those to the electron transport chain in order to produce ATP. So we have glycolysis and pyruvate oxidation, and we have the citric acid cycle here. Just remember your inputs, your outputs, what's going on, how are we carrying those electrons to the ETC in order to perform oxidative phosphorylation. And we also see here that we're giving our electrons to oxygen, which is the terminal electron acceptor. And the reason I'm highlighting that here is because it kind of begs the question, what happens when we don't have oxygen? Well, that's when we perform anaerobic respiration, respiration without oxygen, which is the process of fermentation. So we see here in the flowchart form, when we have oxygen present, we use that to accept our electrons, so we run for our normal processes of pyruvate oxidation, citric acid cycle, oxidative phosphorylation. But when we don't have oxygen to perform oxidative phosphorylation with, we undergo fermentation, so either ethanol or lactic acid fermentation. Now, human cells do not perform ethanol fermentation. Our cells are not making ethanol. They won't make ethanol. We will die if that happens. It doesn't happen. But here, in some yeast and some bacteria, we perform ethanol fermentation. That's how we get alcohol, so go yeast. We perform lactic acid fermentation. So I like to think about fermentation as our last resort. Why? Because fermentation allows glycolysis to run. Now recall that glycolysis produces a little bit of ATP through substrate level phosphorylation, which means that we get some net ATP to carry out cellular processes. And we don't require oxygen at all in this process. But we do require NAD+ as an input, and we make NADH. So if there's no way of getting NAD+ back, we're going to burn through all our NAD+ and be left with a bunch of NADH, and we can no longer perform glycolysis. That's where fermentation steps in. So fermentation is going to take this NADH as an input and release NAD+ as an output by taking our pyruvate product and turning it into lactate instead of oxidizing it to acetyl-CoA. So we regenerate our NAD+ and allow glycolysis to take place. So recognize that this does not generate ATP. That's a common misconception. This fermentation step of taking pyruvate and turning it into lactate, nowhere here is ATP an output. No, it's only reducing regenerating NAD+ so that we can generate ATP via glycolysis. That's what generates ATP. So think about how this might happen. Like in our muscle cells, when we're doing a lot of aerobic exercise, we're using up a lot of our oxygen stores, and our muscle cells then produce lactate as a last resort to keep producing ATP to keep their function. That's when we experience muscle cramps because we have lactate building up. And again, same process in yeast and those bacteria when they perform ethanol fermentation. We're still regenerating NAD+. We just have this extra intermediate of acetaldehyde, which releases CO2 as a byproduct. That's why beer, other products, kombucha, has CO2 in it, because that's what we're producing as a result of anaerobic ethanol fermentation. Big idea here, key takeaway, we're regenerating NAD+. So where are all these processes located? Well, in eukaryotes we have both the cytoplasm and then the mitochondria with a bunch of membranes. So in the cytoplasm, that's where glycolysis and fermentation are going to take place, but then we import the pyruvate product to the mitochondria, and we perform pyruvate oxidation citric acid cycle in the mitochondrial matrix. And then even further within, that's when we perform the electron transport chain, because recall that in order to pump a proton, we need a membrane, so that happens in the mitochondrial inner membrane. Well, in prokaryotes, they don't have any mitochondria. They don't have anything. They're kind of boring. So all they have is the plasma membrane, so that's where they have to perform the electron transport chain because you need a membrane for that. Everything else is going to take place in the cytoplasm because, again, there's nothing else there. So all these processes have to take place in the cytoplasm, and then we use their one plasma membrane in order to pump those protons. I also just wanted to add a quick note about where fats and proteins come into this process. So fats are going to enter into the citric acid cycle in the form of acetyl-CoA, and a part of fats, which is the glycerol head, is going to undergo glycolysis. Now for proteins can be super different because of how the different amino acids are composed. So the amino acids can either enter in the form of pyruvate, in the form of acetyl-CoA, or as intermediate molecules in the citric acid cycle. So my best recommendation is just to memorize this diagram right here and recognize that fats are going to enter as acetyl-CoA, and the amino acids can basically enter in 3 different spots. And now for the process of photosynthesis in plants. So plants can either have, or any organism performing photosynthesis can either have photosystem 1, photosystem 2, or both in the Z scheme. So if an organism has photosystem 1 only, it's going to be a photoautotroph. So it's going to do 2 things. 1, it's going to make ATP, and 2, it's going to have the ability to reduce carbon dioxide into carbon compounds. So I've literally written out the steps of what will happen with photosystem 1. You don't have to try and decode the textbook or anything. Here's what happens. We have light. It's going to come in and it's going to oxidize chlorophyll. So chlorophyll will lose this 1 electron, and this electron is going to transfer to NADP+. It's going to be reduced to NADPH. Now NADPH and NADP+ are just the plant forms of NAD+ and NADPH. Okay, so we have this NADPH. That is the way that we have reducing power in order to reduce carbon dioxide later, because we're building up NADPH. Now chlorophyll, remember, is oxidized, so it has to be reduced in order for the cycle to continue. And it's reduced by a terminal electron acceptor. Commonly in bacteria, it's hydrogen sulfide. So it's going to reduce the chlorophyll so that it can be again excited by light. And so if it's reduced, it's gaining an electron. That electron is going to enter the electron transport chain. And same idea as in animal cells, when we pump that proton, we allow ATP synthase to spin, so we're making ATP. So we're doing two things. We're providing reducing power in the form of NADPH, and we're making ATP. Well, what about with photosystem 2 only? Well, in these organisms, they're going to be phototrophs, so all they can do is make ATP. And so same idea where we're oxidizing the chlorophyll, but then what happens is that because we're losing this electron, we're going to oxidize water. So we're going to get an electron from water. This electron is going to enter into the ETC, it's going to pump a proton, and it's going to allow ATP synthase to spin. So if you look here, there is no NADPH being generated. We are only pumping a proton, so we are only generating ATP. And then that electron is going to return to PS2. So this is cyclic electron flow. Okay, so what happens when we smush these two photosystems together? That's when we get the Z scheme. So the Z scheme starts with photosystem 2. So that light is going to come in, it's going to oxidize PS2. Water is going to be oxidized in order to reduce it. So we take that electron, we give it to the ETC in order to pump a proton across. So now that the ETC has been reduced, I'm going to write that right here, ETC reduced, it has gained an electron, it's going to give its electron to PS1. So then PS1 will be reduced because it has gained the electron. You see that right here where the electron goes to PS1. Well, PS1 is going to do its job, and it's going to reduce NADP plus into NADPH. So we've done two things. One, we've generated reducing power in the form of NADPH, and we've generated ATP here. You can see that because we built up this gradient of protons where we pumped it right here in the ETC. So we have NADPH for reducing power, and we have ATP synthase spinning because of the proton gradient. So I've said a lot about this reducing power in the form of NADPH. Well, what that means is that we can take carbon dioxide and we can fix it into a usable form of carbon, aka we can reduce it. And this is going to take place in the stroma, which is deeper inside the plant chloroplast, which is kind of why it's called the light-independent reaction. Those photosystems have to take place on the outside, obviously, because they're absorbing light in the well, I'm not a big plants person, okay? It takes place further outside of the chloroplast. Anyways, so what's going to happen is that this carbon is going to be fixed in a process known as the Calvin cycle. So this carbon dioxide enters in, and it combines with RuBP, and it gets turned into some 3 carbon molecules via the enzyme Rubisco. So that's that first step right here. Then what happens is that all that reducing power in the form of NADPH arrives and is going to reduce the molecule, and 1, produce NADP+, and 2, produce a sugar. So this molecule has been reduced, 1 of these molecules is going to exit as a sugar, and the rest are used to regenerate RuBP and continue the cycle on. So again, carbon dioxide enters, Rubisco works to turn it into some 3 carbon molecules, those molecules are reduced into sugar, 1 of them exits, the rest regenerates RuBP so the cycle can continue. Now, there are 2 ways to disrupt this cycle. The first is when carbon dioxide is removed, and the second is when light is removed. And we'll see those impacts right here with this different diagram where we actually see the molecules. So without carbon dioxide, I like to think about this in terms of where our roadblocks are going to form. So without carbon dioxide, boom, we stop the cycle right here. So what's going to happen? Everything after carbon dioxide, it's going to spin around until we hit that roadblock. So our 3 PGA molecules right here, they're going to all be used up because the rest of the cycle is going to run until we hit that roadblock. It's going to run until we all make RuBP. So RuBP is going to accumulate, and 3 PGA is going to disappear because we're just running that cycle until we have run out of 3 PGA and built up all the RuBP we can. Now, what about without light? Well, without light is just another way of saying without NADPH, because light is used to generate NADPH. So where is the roadblock going to happen then? Well, right here, right? No NADPH. Well, again, what's going to happen? All these G3P molecules are going to run until they cannot anymore. So G3P is going to be used up. It's going to disappear, and it's all going to be turned into 3PGA, because that's what the last thing before we hit that roadblock is. So 3PGA is going to build up, G3P is going to be used up, it's going to disappear. So again, in terms of disruptions to the Calvin cycle, just think about where our roadblock is going to appear and how the cycle is going to run until it can no longer. So yeah, that was a really fast overview of the metabolism of animals and plants. I hope it helped, and good luck with your studies.